So the next part is to start combining the single cell ATAC-seq with single cell RNA-seq. You probably saw a little bit in the previous slide that after we know some transcription factor with ChIP-seq that has significant overlap with your ATAC-seq peaks, you still want to check whether the transcription factor is expressed or not, which means, like we do care about expression level of the genes. And so in order to really combine the single cell RNA-seq with the single cell ATAC-seq, we need to really kind of figure out a way to say, okay, this cell is roughly that cell type, this cell is roughly that cell type. And in order to do this, the current ATAC-seq or single cell ATAC-seq analysis approach, usually they try to convert the original peak by cell matrix, this count matrix, into a gene by cell matrix. So somehow if you can combine the peaks into every gene has a number, then the ATAC-seq data will be looking almost the same as your single cell RNA-seq, and hopefully they will really overlap with each other and you can make them work. And so the question is, if we know in each single cell what regions are the open chromatin, remember that the peak by cell matrix can be hundreds of thousands of lines long, because like if you look at ATAC-seq data, you usually get hundreds of thousands of peaks. Whereas the gene by cell matrix, you have like 20,000 genes per cell, right? So we need to reduce the total hundreds of thousands of rows into about 20,000 genes. How do we do that? In the early days, people try to look at whether the promoter of a gene is open or not. This will give you some reasonable result if you have very, very different tissues. For example, if you have using a cell line, very different cell lines, you try to mix them together and create a fake, like complex tissue to do single cell ATAC-seq and single cell RNA-seq. I think the promoter openness can give you some, reasonable idea. But for example, if you were to do PBMC, these are all already white blood cells in your body, and all the white blood cells share many open chromatin. At least they are all the same cell lineage. We are not comparing brain with liver and like totally different things. So at that time, the promoter openness is not as good a proxy for openness. As we mentioned, in many cells with similar lineages, the promoter might be open and waiting for the correct transcription factor to be expressed, then potentially looping to the promoter to kick the polymerase out, like to start RNA transcription and polymerase II elongation, right? And so before the gene is even expressed, polymerase II can be already sitting on top of the gene, and that promoter is open compared to the gene that has no chance of expressing that cell lineage. And so just having a promoter openness is not a very good proxy for gene expression. Later on, some algorithms also look at how much you have gene body read coverage, which is to say, if we look at from the beginning of this gene to the end of this gene, any reads that lands in here are going to be lumped together to give to this gene. You can do something like that. Of course, you need to normalize by how long the genes are, because long genes, obviously, you have more chance to have some reads that lands there. And you can do something like that. There are also different implementations. You can smooth the data with the K nearest neighbor cells. Remember, we can use the ATAC-seq data to do clustering. And so if the readout for each gene near this gene in this cell is because it's very, very sparse, you can also look at, say, 10 cells or 50 cells near this cell that's kind of clustered, and like merge all of their reads together to look at how much a gene is expressed on this, how much ATAC-seq read you have near this gene. And so people also do this gene body read coverage. My own group, we have an algorithm called Maestro. It's not published yet. We are submitting the final revision soon. We have an algorithm, this Maestro, which you can get now in this website. we use regulatory potential, which you have seen in homework 4 to assign the transcription factor binding sites to every gene. Basically, if you have this could be a transcription factor peak. It could also just be a TAC-seq peak. We use exponential decay by distance to decide how much peak or read is being assigned to the promoter of the gene. You can imagine if we have the single cell vector, right? So the single cell have these locations as ones. For each location, we just ask how far is it. If it's really around the transcription start site, we'll count it as 1. But if it's further away, it was count maybe as half if it's 10 kb away. But if it's really like 100 kb away, then it's a tiny number. And so we will be able to consider things that are further away, but weighted by the distance for every location that has a 1 in that peak location, and consider whether this gene is expressed or not. And that can give us, no matter how many reads you have per cell, we will be able to get a number on every gene. And so this approach, we will show you later that it seemed to work best in terms of really capturing gene expression. And so once we can convert the peak to cell count matrix into a gene to by cell expression matrix, now then your single cell ATAC-seq data will look like a single cell RNA-seq. Every gene on every cell will have a number. Next gene on the next cell will have a number, right? So then we can use so with single cell ATAC-seq, we have a number of cells. With single cell RNA-seq, we have a number of cells. And we can pretend that these are 2 batches. Remember, with single cell RNA-seq, if you have, say, 2 tumor samples you assume roughly similar overall cell population. In this case, where we are doing single cell RNA-seq and single cell ATAC-seq on the same tissue, the initial same sample, you just split the cells into 2, so they should have similar cell representation. Then the single cell ATAC-seq, we just convert it into a similar like single cell RNA-seq data infrastructure, and you compare with your original single cell RNA-seq, because they should roughly have similar representation, we can use the CCA approach for batch effect removal to merge the 2 cells together, the 2 samples together. Okay, and so with that, you can see here, so the green and red, I forgot which one is which, but one is the single cell RNA-seq and the other is the single cell ATAC-seq. And when you run CCA, they really try to overlap with each other. And then with single cell RNA-seq, we can easily look at the expression in that cluster and then assign the cell type based on the marker gene, which you did with, well, you are doing with homework, well, you did with homework 3. And so now we can transfer the single cell RNA-seq cell identity into the single cell ATAC-seq as well. So you can see how here's the B cells, and these are the T cells, and these are the monocyte and macrophages, and so on. Okay, so basically using the regulatory potential, we can convert the original peaks into now all genes. And once you have single cell ATAC-seq and single cell RNA-seq both in the gene by cell matrix, you can just pretend there are 2 different batches of the same sample. You can merge them together and annotate the cells. So we can show you, for example, this is the Maestro implementation. This is the Surat, SNAC, PATAC, Cicero. They use various approach looking at promoter openness or gene body coverage. But when you look at, look at after you have the cluster, what is the gene measures they have on the cell versus in the single cell RNA-seq versus single cell ATAC-seq, and you ask whether the ATAC-seq gene that this measure agrees with the RNA-seq, you can see actually Maestro gives the best correlations between the ATAC-seq measured gene by gene measure versus the RNA-seq measured gene by gene measure on the same cell cluster. So it's not perfect. I mean, we're only getting, around 0.2 the Pearson correlation. You can see here, this is one example. Each dot is 1 cell. the x-axis is how much you see in terms of gene expression based on the single-cell RNA-seq, and the y for that cluster. And the y-axis is how much you are seeing on the single-cell ATAC-seq for that gene. And you can look at the correlation. I mean, it's from Pearson correlation, we get 0.26. From Spearman correlation, we get 0.45. It's pretty good, but it's certainly not, like a real tool, a single-cell RNA-seq, you get, like 0.8, 0.9 kind of correlation. But this is already a lot better than, for example, looking at the promoter openness or looking at gene body coverage. But that's kind of roughly, we always use this exponential decay to weight all the peaks to a gene. This way we can compare one gene in the ATAC-seq to the same gene in the RNA-seq. Okay, and so with that, I also want to, we might go a little bit over, I hope it's okay with you guys. I also want to mention another algorithm that's developed in my group. It's called ELISA. It basically predicts potential driver transcription factors from any gene sets that you're interested in. For example, if you input a list of differential genes from a drug treatment, a gene knockout, activation of some process, a cell differentiation, anytime you have differential genes, it can't be too few. If you don't have 3 genes, it's very difficult. But if you have, a few hundred differential genes, ELISA, this is a website. It can use all the public H3 K27 acetylation ChIP-seq or chromatin accessibility profiles to select the chromatin profiles that are relevant to your differential gene expression set, and then try to predict which transcription factor that has either ChIP-seq peaks or motifs that are enriched in your epigenetic profiles relevant to your input gene set. And so I want to kind of roughly show you how this is done. So you can imagine, originally you might have one ATAC-seq data. This is say one maybe K27 acetylation. It could also be DNase hypersensitivity profiles. It's kind of a genome-wide profile with many peaks, many reads. We can convert that, again, using regulatory potential into a RP vector, regulatory potential vector. Basically, this gene has a score which is based on all the nearby DNase profile. It's similar to how you do single-cell ATAC-seq, right? So you look at whether this gene has many peaks and close by peaks to my gene of interest, and the peak height is high, and then we'll just add them together. So it has probably high score. Another gene, this is somewhere else in the genome, there are no peaks, nothing, you have a low score. And so basically with every genome-wide DNase profile or K27 profile, we can convert whatever the original data into a gene vector. Every gene will have a score. High means that there are many peaks near the gene, whereas low number means there are almost no peaks or the peaks are really far away, right? So every DNase profile or every K27 acetylation profile is being converted into this gene vector. Every gene will have a score. And now if I am interested in a group of genes that are differentially expressed, as we mentioned, this input gene cannot be too few. If you look at differential expression between developmental stage or some real perturbations, usually you get, you looked at homework 2 or 3, you can easily get hundreds or thousands of genes that are different. And so if you enter those gene sets, it can't be too big either. If you have like 10,000 genes differentially expressed, it's also quite meaningless. But if you have, like 400 genes that are different, we can use lasso. Remember this feature selection. You can select, select the public either DNase profile or the K27 acetylation profiles and give them a different weight, give it a coefficient, so that the weight you give to the first sample on this gene and the second sample on this gene, the third sample on this gene, basically every gene originally has a score, and that gene, that sample has a weight. You give it that coefficient, at the end, all the samples together with the weight, not all the samples, the samples you selected will give you a final score on each gene. And so this is probably some, this sample selected a positive weighted, and those samples are not are selected but having a negative weight. At the end, it will give you, which genes have high score, which genes have a low score. And if this thing corresponds very well with your target gene, that's the right samples you select. And so basically, we are selecting public DNAs or ATAC-seq or K27 acetylation profiles that their gene scores are also kind of representative of your differential gene expression list. And very often with this approach, all the top profiles we selected are related to your cell lineage. Just based on the differential gene, we will know this is blood, that's T cells, that's some brain tissues, and so on. This is just one example. If we have a GATA-6 knockdown, these genes are downregulated. And then we just look at all the public data and we ask, which profiles we select, say some 20 profiles. At the end, our model looks almost the same as original, this input figure. And so that's how we create the epigenetic profile. We say, okay, these are the profiles we want to consider. And the next one is we then examine each transcription factor either with a null motif or they already have ChIP-seq data. Then we ask if in this, so for example, if this is the GATA-6 ChIP-seq and we know where the peaks are, anytime that it overlaps with one of the chromatin accessibility profiles in here, we just in silico delete this region. If we delete this from the chromatin accessibility region and we recalculate the regulator potential of this chromatin profile. And we ask how big a difference would it change? So basically we ask if we eliminate all the GATA-6 binding sites, how much does it change on the regulatory potential of every gene? And we want to see, basically, if it's the correct transcription factor, if you eliminate all its binding sites, from the DNase peaks or the K27 acetylation ChIP-seq peaks, if it's a correct factor, those genes will no longer be differentially expressed, right? So you will see the changes on the regulatory potential if you delete all the locations that have this ChIP-seq data. And if you can really see a big change on those genes you are interested in, that's the transcription factor that's important. And so, if we look at this in a real single cell RNA-seq example, if you only have single cell ATAC-seq data, you can already use this GIGOS to look at the ChIP-seq data that has the best overlap with your differential ATAC-seq peaks. But you can also run ELISA on the differential genes between one cluster and all the other clusters, and then run ELISA to figure out what transcription factors are interesting. And this is one example that we give you. The x-axis is the result from ELISA, and the y-axis is the result. The ELISA is run on the single cell RNA-seq differential genes, and GIGO is run on the single cell ATAC-seq differential peaks. And you can see a lot of the factors, every dot is a predicted factor. And most of them are actually positively correlated. And we are especially interested in those transcription factors that are upregulated because they are higher. They bind to more regions. They also regulate those genes that are unique in that cluster. So these, CEBP beta or SPI1, these are turn out to be the right transcription factor that we know are related to that cell cluster. And the color just indicate how much the transcription factor itself is differentially expressed from gene expression. So you can see, if you look at both the differential genes and also the differential peaks, it provides you a lot of good information about what transcription factor is really important in regulating the genes in that cluster. Occasionally, we do see some factors like FOS1, you only see it in the differential ATAC, but not the differential gene. I think right now, the single cell ATAC-seq data is still very limited. With more and more data, we'll be able to learn this better. But this is kind of a useful thing. If you have any differential gene expression, you even without ATAC-seq, you can now try to run it on ELISA and see what factors are potentially interesting in regulating your genes. Okay, so in summary, basically, once you have single cell ATAC-seq, what are the things you do? You first map the reads, and then you can do peak calling to call the peaks that are mapped. And then you can look at the sample QC and the cell QC to remove the low quality cells. And then you can do this, generate the peak by cell matrix, count matrix. And so these are the remaining good cells you want to consider. And then based on this cell by peak matrix, you can cluster cells that have same rows as being, you see a lot of ones together in the same co-accessibility, basically. You can cluster the cells that have similar open chromatin. You can visualize the result on UMAP or t-SNE. You can also use the non-parametric test for looking at differential peaks. And then you can annotate which transcription factors might be important using either ChromeVar or the SystemDB, using Giggle to look at ChIP-seq that overlap with your peaks. And in order to really integrate the single cell ATAC-seq with single cell RNA-seq, you use the regulatory potential to assign all the ATAC-seq peaks onto genes so that you have same dimension with a single cell RNA-seq. And then you use CCA pretending the ATAC-seq is just a different batch to merge the ATAC-seq cells and the RNA-seq cells together. And then after that, you can further identify the driver transcription factors by looking at the differential genes and use also public ChIP-seq and the transcription factor motifs and chromatin data to help you predict the transcription factors. And of course, you can use the marker genes from the clusters to annotate the cell types. So I want to mention that this is the first time I give single cell ATAC-seq lecture. Both the experimental technology and the computational methods are still fast evolving. So it's very likely that next year we'll see even better technology, even better methods. So stay tuned, okay? Thank you. So I think that's all for today.
Sorry, one question.
Oh, sure.
Okay, can you explain again, what do you mean by like for ELISA, what do you mean by like weighting and selecting publicly available DNAs or K27 ChIP-seq data?
Yeah, so you can imagine the weighting is assigned onto each column. So for example, I'm interested in these genes, right? And if I select so lasso is to help you do feature selection, but it can also give you a coefficient. The coefficient is on each sample. And so finally, this particular score is this pink level on the gene weighted by the weight on this sample, plus this pink number weighted by the weight for this sample, plus this pink number on the gene weighted by the weight for the sample. And you add all of those coefficients together, well, so the level weighted by the coefficient together, that will give you the final number. And so you can imagine this is a regression approach. You are trying to select among many, many columns, a few columns, and give them a weight on each column so that at the end, the final model of the gene agrees with your inputs, because your input data set, you're saying this is 1, the rest are 0, right? And you are trying to select a vector and weight those vectors so that at the end on the genes, you also have these as 1s and the remaining as 0s.
Okay.
So the weight is on each sample, and the score on each sample is the gene you get based on the regulatory potential of on that gene.
So do we basically like weight and select some regulatory scores and combine them together to give a, I don't know, like the average score? Or
You know what? Maybe we should talk after class. I'll stop this recording and I'll talk to you separately. We can stay on the lecture as well.
oh, okay, sure.
I can stop the recording.